Really exciting for you today. So I'm Erin. I'm one of the registered dietitians with the GI division, gastroenterology division. And then we have Allison Borman, our nurse practitioner with our celiac disease program today as well. So during the talk, please feel free to utilize the chat function to share any questions or comments. And Allison and I will be monitoring the chat, and then we'll have time for the Q&A near the end, at which point you can say your questions out loud, or we're happy to read them off in the chat for you as well.
Okay, great. Thanks, Erin. Again, I'm Allison Borman. I'm the nurse practitioner for the Celiac Disease Program. It's my pleasure to introduce Dr. Antonio Santos, who is a postdoc researcher at Stanford University School of Medicine. Dr. Santos has a background in micro and molecular biology with an interest in organoid modeling. This afternoon, we're very lucky to have him join us to share insights from his current research on organoid modeling in celiac disease. His innovative work offers exciting new perspectives, and we are very lucky to have him here with us today. Dr. Santos, I'll let you take it away.
Thank you so much. I would like to thank the Hillary for inviting me to do this. It's a really pleasure for me to be able to give this webinar, especially since it's for the general public. It's not every day that we get to present to the community. And so I'm going to start by trying to share my screen. Let me know if everything looks good. Can you see it?
We can. That looks great.
Perfect. So I'm going to start. Let me just, sorry about this, real quick. Okay. So yeah, so my talk is about a new organoid model that I generated being in the Cool Lab here at Stanford. And it's really a way to recapitulate human disease, in this case, celiac disease, in a petri dish. This is very novel. Normal people try to recreate human disease using a mouse model, which is not great because it's a mouse. Also using cell lines or very incomplete cultures in the lab. So this is a more like complete, the best version of an organoid, of a human model in a petri dish. And I used it to study celiac disease. So this slide is very simple, and it represents the evolution of how we scientists cultivate human cells in the lab. So cultivating meaning putting them in a solution that is rich in nutrients and have them grow and divide. And so we went from 2D cell lines, which were monolayers of cells. Normally, this system is very limited because the cells are not diverse at all. They do not represent the tissue where they come from. Normally, they just represent one cell type of the tissue where they come from. And then in the last 10 years or so, scientists started using a different system. We call them organoids. And these are three-dimensional, so 3D miniaturized versions of the organs or the tissues where they come from. And they are much better than the 2D cell way because they have a lot more variety of cell types. They grow in a 3D way, so the cells are folded, are shaped in a correct way. And because they are much closer to the actual tissue of origin, they can recapitulate new features of the tissue or human disease, depending, much better than 2D cell line. And this slide here is a brief story of organoids, just to give like an idea of what they are and what they represent. The modern organoids start in 2009 with intestine gastric organoids. But there's all sorts of organoids. Nowadays, we can do organoids from any animal. Some people even did snake venom gland organoids, and these organoids can make venom, for instance. But there's all sorts of kinds, brain organoids, mammary gland, prostate, lung, retina, liver. So we are really evolved into a much better system that we didn't have like 10 years ago. And the organoids can be used for many different applications that can help us understanding human physiology much better and then treat disease. We can do fundamental research to find fundamental things about humans and how human cells work. But we can also use the organoids to model diseases, like what I'm going to do, what I'm going to show. And then if we model a disease, we can use the organoids to test drugs. So we can treat the different drugs and then see what is better for the disease, what cures the disease. And these are just a few examples out of many things that the organoids can do. And I just want to remind everybody that before the organoids, this was very, very limited and very inaccurate. So today I'm going to talk about the small intestine because that's the tissue in which celiac disease is manifests. And so the small intestine is characterized by two morphological features. We have crypts in the bottom and then villas on top. These are like protrusions that are on the top of the crypts. And the reason why they are folded like this in protrusions instead of just parallel is to increase surface area so we can get more nutrients out of food. So the small intestine really is made for nutrient absorption. And another two things that I want to show in this slide is that we have two very different compartments in the small intestine. We have the epithelium that realizes the functions of the small intestine. So there are cell types there that absorb the nutrients. There are other cell types that keep bacteria away or kill bacteria. So the epithelium is really like making the function. But the epithelium is only surviving, is only thriving because they are supported by other cell types that are not epithelium cell types. And we call this amalgamation of cells lamina propria. And they are very important because they support the integrity of the epithelium. And very importantly, when I get a biopsy, when celiac patients go to the clinic to get an endoscopy, if they consent to help with research, I will get a biopsy. And the biopsy contains both the compartments that I mentioned, the epithelial compartment, but also the lamina propria. And important for celiac disease is that the lamina propria also has immune cells. So I get not only epithelial cells, but I get all sorts of different cell types that sustain the epithelium, which includes the immune cell. So what I further developed in the lab here at Stanford was a system that consists of an air-liquid interface that allows to grow not only the epithelial cells that I mentioned, but also the lamina propria cells that contain immune cells. So this is a great advancement because the organoids that I mentioned before, they don't have immune cells. So therefore, they cannot be used to disease like celiac disease or inflammatory bowel disease or other examples. They can only study the function of the epithelium compartment. So this is how the small intestinal organoids that I grow here in the cool lab using this ALI or L interface look like. And very interestingly, even just um, cultivating them for 2 weeks or less, they form spontaneous protrusions, the villas that I was mentioning before. And also in this uh, photograph here in the bottom, I also have in green proliferative cells. So the cells not only are recapitulating the morphology of the small intestine, but they're also like dividing, they are increasing in numbers. So it's a system that is thriving. And it's all in vitro, the tissue of the person that it came from. So interestingly, I can grow them for a long time easily. So they are like really happy in the culture. And if I look at the proliferation of the cells, represented here in red, I can see that the organoid, this 3D organoid that has immune cells, is really happy and proliferative. So when I look at the organoids closely by microscopy techniques, we can look at specific cell types. So I see that they have cells that produce mucus, which is important to keep bacteria away. They also produce a protein called chromogranin A that's important to communicate with the enteric nervous system. They also have cells that contain lysozyme that's important also to keep pathogenic bacteria, bacteria that makes us sick. And as I said before, we also have the other cell types that are not just the epithelium ones. We have all sorts of different cells that do different things, like blood vessel forming cells. We have neurons in there. We have fibroblasts that are important to make growth factors or important for the cells. And importantly, to study a disease like disease, my organoids also have immune cells that are represented here in green. And this is very important because this is basically the first organoid model that is not coming from a tumor that contains the epithelial cells that make the function of the small intestine, but they also have immune cells. And therefore, we can use them to study more complex disease like celiac disease. And like those immune cells from the organoids, I see that I have a very diverse population of immune cells. We have immune cells that kill pathogens, that kill viruses, that kill cancer. We have others that make antibodies. We have others that um, prevent inflammation. So depending on the situation, depending on what's going on in our small intestine. And this is just a confirmation again by microscopy techniques. We can really localize them in the organoids. So we can see that cells that eat other cells or eat bacteria. In red, we can see cells in green that prevent us from getting cancer, for instance. We have cells in green on the right-hand side that make antibodies. And we also have the right localization. So the same way that they localize in the real tissue of the person, they also localize correctly in the organoids. And this is represented here by the green separating from the red cells. So now I'm going to talk about celiac disease, which is the disease that I modeled using this system. And this is an image of how a small intestine tissue looks like on the left-hand side. That's the normal, that's how it's supposed to look. But when a celiac person eats gluten, like bread or pizza, they get destruction of the epithelium. The medical term for that is villi blunting, and that basically is epithelial cell death. The disease prevalence is around 1%. It's probably more, it's probably underestimated. There is constant inflammation, there's constant like immune attack in the small intestine, and that's why there is cell. And because of that, the epithelium is not there. They cannot do the normal functions. So normally people lose weight, they have diarrhea. So there's all sorts of problems. And all celiac patients have a genetic predisposition. So that's why we do genetic tests on them. And so this is the kind of thing that I do with my organoids. So I get the endoscopies from the clinic when a patient is making an endoscopy, and then I make my culture, make my culture. I wait for 1 to 2 weeks, and then I treat them with either gluten. So I'm giving gluten to the organoids in vitro, and the most common gluten form is gliadin. So every time you hear me saying gliadin, I mean the control. I mean the gluten, and then I also have the clip control. The control is not supposed to do anything. It's just a way to compare. And so this is what happens when a celiac patient eats gluten. They have gluten peptides that go to the lamina propria, they can cross the epithelium to go to the lamina propria, and then the gluten gets sent by specific immune cells. We call them T cells, and these T cells, they only recognize gluten. So all celiac patients have these nasty T cells in them, and they are the ones that sense the gluten, and they start doing a lot of things that are really bad. So one thing that they do is that they tell other immune cells to start making antibodies that attack our own proteins. And one thing I do see in the organoids is that the organoids can make antibodies. This is a graph representing that. So the red are the organoids that come from celiac patients versus the gray, which is organoids that come from control patients, non-celiac patients. And the celiac organoids, they are making antibodies in vitro, in a dish, which is really cool. And then these cells are really neat, these gluten-specific T cells. They do all sorts of things. They um, start activating cascades of inflammation. For instance, they tell the epithelial cells that are remaining, the few that are still there, to start making a protein that we call IL-15 interleukin. This is a pro-inflammation, so this protein is going to increase the inflammation. And they do all sorts of things. They increase all cytotoxic markers, things that lead to cell death. They increase chemokines, which is also pro-inflammatory proteins, cytokines, and they activate the immune system. So these cells are able to tell the other cells to get activated and to start attacking the normal tissue. And that's why celiac patients, when they eat gluten, they get all this inflammation and cell destruction. So eventually there is cell death, epithelial killing. So I'm showing in this video dead cells in red. And this is what happens to organoids from a celiac patient after I give them gluten. Look at how all the live cells are turning into dead cells. There's literally holes opening after I give them gluten. So this is really like the tissue destruction that I was mentioning happening in vitro. I also developed a cell death assay to actually read this, making videos all the time. I can always look directly by a technique that we call FACS. And what happens is, for instance, if I block the gluten sensing by those really mean T cells, I get more death, more destruction. But if I block the sensing, I don't have destruction anymore. So this is a good way of like showing that the organ is what happens when a celiac patient eats gluten. So this leads to allows for a platform that can be a platform for new discoveries. So a discovery that I did that I'm not going to have time to mention the details was that IL-7 plays a role. This is another pro-inflammatory interleukin. It plays a role in celiac disease, and I've done that by doing cell death assays that I mentioned. So I can reverse the gluten-induced cell death by blocking IL-7. And for instance, IL-7 alone can kill the epithelium. And so what I found out by doing all sorts of experiments in the lab was that IL-7 and gluten sensing, they are in the same pathway, the same cascade of events, which was not known. This is a organoid-driven discovery. And I confirmed that by understanding by microscopy that the gluten can induces the organoids to start making more IL-7 as compared to non-celiac controls. This is the quantification of that. And then when I look at the actual tissue, not the organoids, of people that stopped eating gluten versus people that are still gluten, this is the actual tissue, not the organoids. You can see the interleukin-7, my discovery, in red. And you can see that people that are still eating gluten have a lot more of this pro-inflammatory protein as compared to people that stopped eating gluten. This is the quantification. And so I came to a model, a working model, in which I believe that gluten is being sensed by those really nasty T cells, and that leads to increased production of IL-7, which leads to the killing of the epithelium. And my paper is published. It has been published in a journal called Nature. It's available, so anyone can go there and read it. It had a lot of impact already because they have immune cells, so it allows to study complex diseases like celiac disease or inflammatory bowel disease. There is like review articles that are talking about my paper, and so this is really cool because we are now raising an antibody together with Stanford's Innovative Medicines Accelerator to try to go to clinical trials. So this is really like started with celiac organoids, and then by studying them, I made a discovery that IL-7, which is pro-inflammatory, is involved in disease. Then we are now creating an antibody together with IMA at Stanford. And then this is ongoing. And then potentially, if everything goes well, we can go to clinical trials to try to treat celiac disease. And this is my acknowledgement slide. This is the team that I work with that helped me a lot. And now I could take questions. Sorry if I went over time.
That's really fascinating work. Thank you so much for sharing.
Thank you. Of
Uh,
course.
Leah, I don't see questions in the chat. I don't know if you're seeing questions.
I don't. No, not yet. So if anybody has some questions, go ahead and feel free to put them in the chat or go ahead and start talking.
We do have one question. It's oh, they're coming in now. Hold on. One question is, do organoids self-organize?
Mm-hmm. Yeah, they spontaneously self-organize into the tissue where they come from. We call them mini organs. So it's like a mini version of the tissue of origin, and we don't completely understand how they know what they are supposed to be. That's more like a stem cell question, but it's an interesting and it's an open field. These organoids are also going to not just allow us to study complex diseases like celiac disease, immune-driven diseases, but it will also like teach us a lot about how organs form. That's another way of using the organoids. They have multiple applications.
Another question is, how well accepted are organoids for clinical trials?
How well accepted are they for?
For clinical trials, or I guess, are they commonly used in clinical trials?
So, for instance, our strategy is the IMA is gonna create an antibody against IL-7, and then I'm gonna go back to my organoids, and then I'm gonna test the antibody if the antibody can prevent activation of the immune cells and then prevents the destruction. So I'm going to do that first in the organoids, and then if that goes well, we can start all the clinical trial phases, which has like a safety phase first, and then a small group first, and then eventually more people and approval. It's the very beginning, but it's still very exciting because potentially we can have a unique way of treating the celiac symptoms if the celiac person eats gluten. So it would be a way for someone that is celiac to eat gluten and not have problems.
And I don't think there are any IL-7 drugs on the market right now. Is that right?
Yeah, so we are going to generate the first anti-IL-7 antibody. But we know that some people have tried the body against the receptor of IL-7, that has been done for inflammatory bowel disease, and they are fine. So we think that this is going to be a very safe antibody to use.
Okay, really interesting. Another question from Anna. Can you please tell us a little bit more about how your discoveries could lead to patient treatment and what those treatments could look like in the future?
I'm sorry, I'm reading the question. So yeah, a lot of the discoveries that about human physiology and human disease were made in mouse models, and 9 out of 10 of those discoveries led to drugs that did not work in humans. So, so we know it's probably going to be better than a mouse model, that we can make discoveries that are probably going to be much closer to or much better at treating human disease as compared to discoveries that can come from mouse models.
And I'll read the next question because I'm not sure everyone in our audience can see them. But from Chris, a question about personalized medicine. Could you take a patient's biopsy and use it to create a customized treatment for that patient?
Exactly. So that's a great question. So there is patient-to-patient variation we see using my organoid system, but also in organoids that are more simple, that only have epithelium. And one direction that is for sure going to happen is to test drugs in the organoid first, and then see which ones work, and then go to the patient where the organoids derives from, and use that is going to be very relevant for tumors. We can also create organoids from the tumors, and then we can test chemotherapy in the organoids and see which chemotherapy kills the tumor, and then go back to the patient and say, or to the doctor and say, this patient is going to be much more susceptible to this drug than rather than another one. So that's going to happen. It's kind of already happening with special patients that um, give a donation to the lab.
Mm-hmm. From Carolyn, can you say more about what the treatment in the trials would actually consist of?
It would be probably an injection of IL-7 antibody. It would not like an injection, more like one of those pen things that are painless. And then the person probably could go and eat a lot of gluten without feeling bad or having problems. Some people don't have a lot of symptoms. They don't feel anything. But when the doctor goes and looks at the biopsies, it's really not good. That also happens.
I'm sure it's hard to answer this, but do you have some prediction about whether or not this could lead to an alternative treatment where patients could eat an unrestricted diet, potentially?
So say that again, sorry.
Do you think that in the future, this type of treatment could lead to a truly alternative treatment to the gluten-free diet, where patients could eat an unrestricted diet?
I think so. My only comment about that is some people have been on a gluten-free diet for so long that they are probably not going to want to change. People definitely will. It's more like a 50. And yeah, especially if the antibody is safe and doesn't have any sort side effects, a lot of people will like to go and have cake on a birthday party as compared to no.
Yeah, absolutely. Absolutely. From Nadine, do the antibodies cause the damage or epithelial cell death? Do the antibodies cause the inflammation from gluten exposure?
Thank you for that question, Nadine. I think just to clarify, I think Nadine is talking about those autoantibodies that are generated after gluten consumption from a celiac individual, not the antibodies to treat it, not the IL-7 antibody. So that's a great question. So if the autoantibodies are pathogenic is known. So that's a question that we want to address using these celiac organoids. It's not known.
Okay. And then also from Nadine, are you able to regrow the entire villi, including the microvilli?
Right now, not really. So it's only the microvilli. So it's not the villi, the complete villi. But I think if we'll find a way. And the reason why I say that is because if we put these organoids inside inside a mouse and then take them back out, they will have much bigger and complex villi than the microvilli. So the mouse is providing some sort of growth factor that allows to create complete villi, real villi from the organoids. We just need to figure out which growth factors are.
Okay, and then we have a really interesting question from Marina. She noticed that your organoids are not from control patients or those without celiac disease. She said,
No,
oh, go
I
ahead.
also have from non-celiac patients, but for this presentation, I showed mostly the ones that come from celiac patients and the effects. But we always have non-celiac controls for everything, making sure and we don't see any gluten-induced effects in the organoids that come from non-celiac patients.
Okay. She had some questions about if you've noticed any differences in how the celiac organoids establish their growth kinetics, et cetera, compared to controls.
Great question. Yes, normally the celiac organoids, because if they come from a celiac that is still eating gluten, or it's like being not having a very good gluten-free diet, like cheating a lot, we see a lot more immune cells because there is like a baseline inflammation going on in that patient. But if you look at organoids that come from non-celiac control or from a celiac patient that is not eating gluten, that is really good at following the diet, we see a lot less immune cells. So the immune cells are definitely, there is a big variability depending on what's the status of the disease.
Okay, and then from Vanessa, has the role of IL-7 in celiac disease been validated in human studies?
No, it's completely a discovery. So I validated it after the organoid discovery when I did the microscopy of IL-7 in the actual tissues, not in the organoids. So this is new. This is new stuff. So in my paper, I discovered and validated it, and now we are trying to help the celiac community with the potential treatment with these antibodies.
Okay. Her second question is, how do you account for the gut environment, including microbiome, in these organoid models?
So yeah, great question. So there is many things that can be done. So this is has just been published. It's very new. It's a new system. So there is many things that we don't know yet. And one great question would be the microbiome. For sure, the microbiome is implicated in at least some uh, autoimmunity, some immune-driven diseases. I can talk about inflammatory bowel disease. That is thought to have a very important microbiome component. So one next step would be to add a microbiome component to these organoids and see how that interacts with the immune cells and the epithelial cells, etc.
Great. And then, yeah, from me, I mean, speaking of next steps, what are the next steps in your research?
We focus a lot on the T cell component of the celiac disease organoids. We are now going to be looking at the B cells, the ones that make autoantibodies. We are going to try to answer if the autoantibodies are pathogenic, if they are themselves doing something. So that would be done, for instance, by taking autoantibodies from celiac uh, and organoids and then putting them in non-celiac organoids to see if there is destruction. That would show that they are directly inducing destruction. We want to know if gluten consumption increases the formation of cells that make an autoantibodies, which would be the plasma cells. So I'm going to shift our gears to the other uh, lymphocytes population. So that's for the near future. The more like the future would be to model other diseases like inflammatory bowel disease, which is much more complicated than celiac, unfortunately. Um, and the Cool lab already is doing a lot of cancer organoids, same technique, and also other diseases that have an immune component, like infectious diseases. So we have lung air liquid interface organoids that we have infected with SARS-CoV-2. Same with brain. So the great thing about this system is that it's novel. It's really almost just the Cool lab. In terms of the small intestine, it's just me. So I'm the only person in the world doing air liquid interface in small intestinal organoids. So there's like a lots of avenues, lots of possibilities. So I could answer that question. I would need like days to answer that question.
Well, I think it goes without saying that we are all really glad that you're doing the work that you are doing. Yeah.
Thank you. And yeah, thanks for the most important people, which were the patients that consented to give tissue for me to do these studies. So thanks to them. Without them, I couldn't have done any of this. And we are happy to at least try to help the celiac community back, potentially with an alternative treatment to the gluten-free diet.
Mhm. Yeah, I think especially in pediatrics, it's really exciting that, some of our youngest patients at some point in their lifetime will have an alternative treatment is very exciting.
Yeah, I agree.
Erin, I see you're off mute. Did you have any questions?
I did actually. I noticed, or I think I read in your paper that you mentioned there could also be possible implications for diagnosing celiac disease, with some of the things you've discovered. I was wondering if you could talk about that a little bit.
Yeah, so for instance, one way that the clinicians um, diagnose celiac disease is to measure the autoantibodies that are in the circulation of blood, in the blood, celiac patients. Um, my organoids also make those same autoantibodies. So by using the same technique, the same kit that they use in the hospital, if I instead of the blood, if I take the solution where I grow the organoids in, the media, if I take that, I can see autoantibody production. So that's direct way of diagnosing it. I can also look at substruction, like I showed today, and see if the organoids coming from a potential celiac individual, if they are reacting to the gluten in a destructive way. That's another way to diagnose. So basically, all the different effects that I showed during my presentation can potentially be a way to diagnose, especially if we combine them. Because some patients, for instance, don't have autoantibodies in circulation, but they have tissue damage or vice versa. So this could definitely be a way by combining by looking at different effects, we can maybe more accurately diagnose a celiac.
And then thank you for answering that. Very interesting. And it looks like we have another question from Jennifer. She says, in what ways can organoids potentially replace the use of animals in celiac research?
Oh, 100%. I think that's after my publication, I don't think anyone would use a mouse model, considering that I have a human model that is in vitro, can be challenged with, can be experimented with. uh, why would we continue using a mouse model? Uh, and yeah, mouse model is also controversial. There is only one that people kind of trust, but it's very controversial. It doesn't work very well. So my system is a human system, not a mouse, and it's a better way of looking at celiac. Mice mice don't have celiac.
All right. We're happy to take any last questions in the next few minutes. While we're waiting to see if any other questions come in, Antonio, how did you first become interested in celiac specifically?
I wasn't. I had I had just developed a system in which this system, this air liquid interface system, and I was showing in a consortium presentation that not only I had epithelial cells there, but I also had immune cells in it. So this is like a human system that does not come from a cancer. It comes from normal tissue that can have immune cells in there. And the professor came to me afterwards, specifically Professor Elizabeth Mellins, that passed away recently. She suggested that I could model celiac disease using my immune-containing organoids. And so I did.
Great. We do have another question from Vanessa. In your model, gliadin deamination occurs in the lamina propria, implying that gliadin peptides traverse the epithelial barrier. Could you elaborate on this process in your system? And could organoids detect disruptions in gut barrier integrity?
Yeah. So quick answer is yes, but it's in a different way. So I didn't show any of that in my presentation today. So what happens is that my organoids are embedded in a matrix that is the collagen matrix. And so if you could see, my presentation is still on, right?
Yes.
If you look at those celiac organoids, that pink area around the spherical organoid, that's collagen. So when I treat the organoids with gluten, for instance, they have access to all the cells there. There is no barrier. And I don't think this is a limitation. I think it actually makes I don't have to worry about the barrier because it works anyway. But one fascinating thing that unfortunately I don't have slides here to show that is if we remove the collagen by the usage of an enzyme that breaks down the collagen, there is a transversal of not only the cell types, but also the polarity. So we have organoids that are floating, that not embedded in a collagen matrix, and they have epithelial cells on the outside and all the other cell types on the inside. They invert. And so when we treat those floating organoids that now have an epithelial on the outside that is polarized, that probably has a barrier that can be tested. If you want to know more, you can send me an email. It's a little complicated, but yeah, in theory, yes, but changing a little bit the way I did things that I showed you.
Thank you. And thank you for this great presentation. I appreciate it.
Oh, thank you.
Yeah, by the way, everyone is welcome to come off mute if they want or turn their videos on if you want to ask questions. In the chat, we do have a very nice thank you from Carolyn. She said, I just wanted to say thank you. As a grandma of a child with celiac disease, I'm so grateful that there is reason for hope.
Thank you so much, Caroline. I really appreciate your comment.
Well, I think we may be winding down on questions, but Antonio, we're so grateful that you joined us today. This was really fascinating and exciting work.
Thank you so much for the invitation and for all the questions and for the moderation. I really appreciate it. for me, it's a pleasure to help, or at least try to help the celiac community.
Yeah, thank you so much. Thanks
Thank
everybody
you.
for attending.
Yes, thank you.
And yeah, thanks for everyone for attending.